in clinical systems biology, building models of the brain and cascades of neuroinflammation to try and come up with novel treatments and diagnostics for illnesses with a neuroinflammatory component. Back in 2024, I was finally offered a position back here in Canada where I had the chance to merge my quantum biology background from my graduate degree with my study of neuroinflammatory illnesses. And that allowed me to form the quantum neurobiology group up in Canada about 2 years ago this May. And so I was awarded a Canada Research Chair. And from that point, I've been trying to use quantum biology to try and understand and treat neuroinflammatory illnesses. So what is a neuroinflammatory illness? Neuroinflammation is inflammation in the brain. So we're all familiar with inflammation in the body. If you bang your arm or elbow, we're familiar with the bruising that occurs, the swelling and the warmth that can occur accompanying that. The brain has its own inflammation response, but because it's a protected compartment, it's separate from regular immune response, so we call it neuroinflammation. What accompanies neuroinflammation, there's a lot of biological processes that are accompanying that inflammatory response, but two main ones that my research lab focuses on. One is an imbalance of metal ions in the brain. So things like iron and copper can go, the concentrations can go out of whack within the brain, causing some havoc. As well as there's an increase in reactive oxygen species, which are oxygen molecules that are scavenging electrons, and they can cause damage to proteins and lipids. So it's something that you'd like to stop in the long run. This damage can be, the generation of this neuroinflammation can be either physical, like a head trauma, banging your head, a concussion. It can be chemical, like exposure to neurotoxic agents, or it can be biological, like exposure to some sort of virus or bacteria. When it's short, so if it occurs over a very acute period of time, it is beneficial and it is something that your body needs and something that you want to occur because it gets rid of that damage and helps restore your body to a natural way of behaving, a natural homeostasis. But when it is long-lasting, the neuroinflammation can lead to long-lasting damages. So when it's chronic, it can lead to damage overall because of the buildup of reactive oxygen species and this long-lasting imbalance of metal ions. One of the things that can occur is specifically in neurodegenerative diseases is that imbalance of metal ions and the reactive oxygen species can start to damage the protein structures in your brain, which eventually lead to the structures that are holding up the brain cell to start to fall apart and die, like in Alzheimer's disease or Parkinson's disease, eventually leading to the neuron withering and being removed. And so the approach of our lab has been trying to come up with novel ways to diagnose this neuroinflammation, as well as novel ways to try and treat the neuroinflammation. And specifically, we're looking at mechanisms based on quantum biology. And so this is focused on light-based therapies, which I will not be talking about today, but which my postdoc, Leah Kassab, will be speaking of right after. But I'll be talking about some of the stuff we're going to be looking at based on magnetic field effects. So magnetic field effects in biology, there's a known effect of magnetic fields in biology, and most of biology can be explained, magnetic fields effects can be explained by standard biochemical and thermodynamic principles. But when we're talking about weak magnetic fields, that's when it starts to get a little bizarre. We heard from Dr. Jones this morning where these weak magnetic fields, they're too low in energy to overcome thermodynamic principles from a classical standpoint. And so we have to come up with alternate ways of describing this kind of behavior. That's exactly what I just said. It goes beyond classical frameworks. But despite the fact that it seems to go beyond classical descriptions, there's a large reported body of work that consistently shows magnetic field effects based in biochemistry at the level of cell behavior, as well as up into animal behavior and so forth. And so we're going to go through some of this stuff. First off, we heard Dr. Jones talk this morning about the avian compass, magnetosensing in the European robin. And this is kind of where this idea of radical pair mechanisms and quantum biology to explain weak magnetic fields in biology started to come about. So as we heard earlier, a radical pair-based mechanism is when you have some sort of chemical reaction up here at the top, And during that chemical reaction, there is a splitting of electrons such that they're shared between two chemical partners, in which case the electrons can have anti-parallel spin, which is known as a singlet state, or one of three forms of a parallel spin, which is known as a triplet state. And depending on how those are formed, and depending on the dynamics that are involved, this singlet radical pair or this triplet radical pair can oscillate back and forth in some quantum superposition. And that can be influenced by external factors such as weak magnetic fields, such that it can force it to stay in the triplet state or stay in the singlet state for longer periods of time. Now, the singlet state, according to physics, it easily recombines and forms your chemical end product. And so you have a quick resolution to that chemical reaction. Whereas in the triplet state, it's not preferred. It's a forbidden physics transition. And so either it will stay in that triplet state and eventually those chemical partners will drift apart and you'll get alternative chemical end products, or it will take a very long time and eventually come back to that original end product. But the rate at which it happens is much longer than the singlet state. So effectively, you can control the speed of these rates by the presence of a weak magnetic field. And that is all the stuff that I just said there. And so in the bird's eye, what it was believed in this avian magnetoreception is that within the eye, in the back of the eye, in the retina, there are these rod cells. And in the rod cells, in between the membranes of the rod cells is where this cryptochrome molecule is embedded. And within cryptochrome, you have the flavin based molecule, as well as a chain of tryptophan molecules. And when this is excited with blue light, it forms a radical pair that can become sensitive to this magnetic field. And then that change, whether that chemical reaction or proceeds or it does not, will cause a cascade, which will eventually cause the neuron to fire or not, sending signals back up to the bird's brain to determine whether it sees the magnetic field or not. This is an active area of investigation. There's still a lot of open questions, but this is the general consensus of generally how this idea would work. Now, beyond avian magnetoreception, there are some other areas where magnetic effects can occur in biology. The first natural place to look is circadian timing. So circadian timing or circadian rhythm is based upon, or it involves the role for cryptochromes in mammalian cells or in animal biology. Now, cryptochromes are an evolutionary conserved family of proteins, part of a larger family of cryptochromes and photolyases. And there's multiple different forms depending on which organism they're involved in. So there's certain ones in Drosophila, there's certain ones in other insects, there's ones in humans, there's others in birds. And depending on which isoform of cryptochrome is being involved, depends on generally what kind of function and what kind of behavior it has. So overall, it is understood that these cryptochromes are photoreceptors, so they respond to blue light, as well as UVA radiation, and can become responsive to exposure to that light, as we heard from in regards to the avian magnetoreceptor. It is generally understood that these are all flavin binding proteins. So in general, it is understood that they bind FAD as a molecule, although this is somewhat questioned in certain isoforms of cryptochromes. So I'll just put that note there, that it is believed that potentially some of these cryptochromes may be vestigial flavoproteins in that they don't bind flavin with a very, very strong degree. Whereas something like cryptochrome 4, which is in the bird's eye, does bind strongly to FAD, indicating that it would be a strong magnetosensor, whereas certain other forms that don't bind FAD strong would not be a magnetoreceptor. But again, this is an area that's actively being investigated. It is definitely understood that cryptochromes, especially mammalian biology and animal biology, helps regulate your circadian rhythm. And again, as we discussed earlier, that these are candidate molecules for being involved in magnetosensing. Again, these are found in all insects, vertebrates, as well as plants. And so potentially, if there are all cryptochromes being sensitive to magnetic fields, you could potentially affect all of these life forms with some sort of weak magnetic field effects. And I believe people are actively looking at all of these areas. So cryptochrome 4 or Cry4 is the one specifically that is implicated in bird magnetoreception in the bird's eye. Is that me? I don't know what that is. In superposition. Is there someone on Teams by chance?
Yeah.
If someone is on Teams, could you please mute yourself? Specifically, mammalian Cry1 and Cry2, as well as Drosophila, Cry act as these core components for biological circadian rhythms.
Can you mute, just turn the sound?
Good? Okay. And no worries. Okay, so because these cryptochromes, these alternate isoforms of cryptochrome are involved in circadian rhythm, this suggests potentially if they are sensitive to magnetic fields, that our circadian rhythms could also be sensitive to magnetic fields. So people have studied this using knockout models in flies and other animals. And they've shown, for example, here that the period of the circadian rhythm is sensitive to weak magnetic fields. So this is some work by Yoshi et al. And it shows that depending on the strength of magnetic field applied, you can get an increased length of the period of your circadian rhythm in Drosophila. They've also shown that if you go and you specifically knock out the cryptochrome molecule and then apply the magnetic field, that you will not get that effect. But if you do have the cryptochrome there, with additional changes, you can get an increased period over time, indicating that change in circadian rhythm is due to both cryptochrome being present as well as the presence of the magnetic field. Other studies have supported this. There's been other work in other insects, so things like cockroaches. People have also done similar studies in humans, which find similar results. However, despite a body of evidence suggesting that this is the case, there are also still contradictory evidence where other studies have done similar approaches and not found any effect. And this, as we go further within this investigation, we'll see that this is kind of a theme in that certain experiments will produce an effect, whereas other experiments will not. And I will advocate for standardization of approaches as we go through this to make sure that when we're comparing studies, we're comparing apples to apples and not apples to grapefruits, okay, over time. Okay, so beyond circadian timing, things like cellular responses also seem to have a response to weak magnetic fields. So in general, weak magnetic fields seem to have an effect on observed cell cultures. So, for example, in human leukemia cells, it's been shown that weak magnetic fields can affect the expression of transcription factors, which are involved in gene regulation within the cell. It's been shown that kinase activity in lymphoid cells is also affected by weak magnetic fields. Calcium levels in satellite cells can be altered by weak magnetic fields. And then finally, things like reactive oxygen species levels can be affected in amniotic cells. Now, this is interesting because reactive oxygen species is a different beast than cryptochrome. So up to this point, we've talked about weak magnetic field effects that are primarily centered on one candidate protein, which is the cryptochrome molecule with its cofactor flavin adenine diphosphate. And so in this case, we're talking about a different molecule based on ROS. And this ties back to the stuff that we're interested in my lab based on damage caused by reactive oxygen species. Now, reactive oxygen species also form radicals, specifically in their name, is that when they are formed, they have a single unpaired electron. And that fact that they have a single unpaired electron makes them very hungry to steal electrons from other locations. And when they flood into the body, they can move around and interact with proteins and lipids and steal electrons from those other locations in order to pair up their unpaired electron. So one that can cause damage, but in that process, they can then themselves set up forms of radical pairs. And so there have been studies that have theoretically modeled this, and therefore it is a different mechanism by which magnetic fields can interact with this biological system. So there's a famous study by Wendy Bean's group that was done a few years back, where they were looking and testing magnetic field effects on reactive oxygen species. And their preferred method of approach is using a planaria model. So planaria is a long flatworm, and this thing is microscopic. You can look at it under a microscope and see it moving around. But it's got a unique behavior that is, if you sever it, if you cut off its head or if you cut off its tail, that it will recruit stem cells and it will regrow itself. And it will regrow itself such that if you cut it completely in half, the head half will regrow a tail and become one planaria, whereas the tail half will regrow a head and it will become a second planaria. And so it forms complete regeneration of its body over time. And so they did experiments where they cut intact planaria and looked at the time it took for them to regrow sections of the planaria and applied weak magnetic fields during that period of regrowth. And so what they found was in that anterior growth, when they were cutting off the head, they were looking at time at which it took for the head to regrow. Depending on the strength of the magnetic field applied, the time at which it would grow would decrease. So at very weak magnetic fields, it would have no effect. If it got a little stronger, it would have a much shorter period. But then if they increased it even further, it would, again, go back to taking longer. And so it seemed to be a very field-dependent effect that over time, depending on the strength of field applied, you could increase the, percent of entire regeneration. So you could decrease the amount that was regenerating and then increase it depending on the amount of field that was applied overall. They also found the same, that if they were doing it on the posterior end, that if you cut off the tail and looked at tail regrowth, they found similar results. They also went then and looked specifically at the levels of ROS, depending on the field supplied. And what they found is that the amount of ROS recruited into those stem cells to trigger the stem cell differentiation and growth was also field dependent, leading them to conclude that the regrowth of stem cells was both field dependent and which correlated with reactive oxygen species. And this led them to conclude that it was a reactive oxygen species driven process as opposed to something like a cryptochrome driven process. Similar results have been observed in other cell types. So there's been work looking at neurogenesis. And again, they have found that it's been dependent on weak magnetic fields, as well as they have found that it is tied, it at least correlates with levels of ROS. So there seems to be some sort of relation there with reactive oxygen species and the weak magnetic field. Whether that is tied through an intermediate of cryptochrome remains to be seen. But it does lead us to question that if there is this other potential mechanism beyond cryptochrome, are there other potential mechanisms that could mediate weak magnetic field effects? What other things are there beyond cryptochrome that can drive weak magnetic field sensitivities in biology? So one of the areas that I'm interested in is particularly, like I mentioned, the imbalance of metal ions within the brain. And so this naturally leads to looking at things like metal isotopes, or specifically metal isotopes that change their nuclear spin. So in this radical pair process, we had mentioned that this oscillation, or this switching between the singlet radical pair and the triplet radical pair, is sensitive to external effects like weak magnetic fields. It is also sensitive to the presence of nuclear spins. So if there's a spin, a nuclear spin on an atom nearby this radical pair, it can change the way that those dynamics take place. Now, nuclear spin is a purely quantum property. So we're not talking about electrons at this point, which we were talking about in the radical pair. We're talking about the spin of the nucleus of an atom. And so the spin of the nucleus changes whether you add a neutron or remove a neutron. It changes the overall spin of the nucleus, but it doesn't change any of the traditional chemistry. The electron number stays the same. The element stays the exact same because the protons stay the same. And therefore, it is the same chemical element. It has a slightly heavier or lighter nucleus, depending on if you're adding or taking away neutrons, but it changes its response to magnetic fields because that nuclear spin is changing. So isotopes with the same element can have different nuclear spins, and it offers a way that isotopes can affect this biochemistry that's beyond classical biochemistry. So classical biochemistry cannot explain nuclear isotope effects in biology or in biochemistry. However, there is evidence of nuclear spin effects in biology. One of the most well-known ones at this point is lithium. So lithium is a monovalent ion, and it's been shown to have an effect on behavior and neuron function. Primarily, it is best well known for being a treatment for bipolar disorder. So bipolar disorder, the common treatment is lithium. It's elemental lithium that you take, and in normal, natural abundance form of lithium is made up of two isotopes, lithium-6 and lithium-7. Lithium-6 and lithium-7 differ in that they have a different nuclear spin. And it's been shown that if you purify lithium and different purified forms of it in animal studies, you can get differing behavioral effects. For example, the one that is most often cited is a difference in rearing behavior of newly maternal rats. So they've just given birth to their offspring, you feed them lithium, and the way that they treat their rat pups is different whether you give them lithium-6 or lithium-7. So in this case particularly, if we look at the nursing, a control group, you have an average amount of nursing. The lithium-7 group, or the natural lithium group, has an infrequent or short duration of nursing. So this is primarily lithium-6, whereas the purified lithium-7 shows, that's natural lithium. The lithium-7 is short and infrequent, whereas the lithium-6 is very frequent and long duration. And so it seems to be a behavioral level effect, but it's based on only a change in the nuclear spin of that lithium. One thing I will note, which caught me off guard, is that if you're taking lithium, it reduces the amount of cannibalization of the pups by the animals, so that it's not all bad. So interesting. It's an interesting effect of lithium. It's also been shown that for some reason the brain seems to uptake a preferred form of lithium. It seems to uptake lithium-6 more strongly than it does lithium-7. So there's another study here where they gave purified lithium-7 and looked at the uptake within the brain. And then they gave a mostly purified form of lithium-6 with a little bit of lithium-7 in there and found a statistically significant increase of lithium-6 within the brain compared to lithium-7. It's been shown that if you induce hyperactivity effects in rats by giving them ketamine, that there's a differing effect on whether they receive lithium-6 or lithium-7, and you can reduce the hyperactivity by giving them lithium-6, whereas lithium-7 will not reduce that hyperactivity over time. It's also been shown, and this is more recent work, it's up on the archive right now. This is work out of Zoya Leonenko's group at the University of Waterloo, and they've shown that hippocampal activity, electrical activity, is differential between lithium-6 and lithium-7, with lithium-7 suppressing activity, whereas lithium-6 enhances that activity. So this is very interesting results, but something we should pay attention to as we go further in these lithium studies. However, other results show no lithium isotope effects. So trying to look for the mechanism by which lithium drives these behavioral processes has been a lot harder than observing these behavioral processes. So there's no lithium isotope effect at the level of cells on GSK3 beta kinase activity, cell viability, mitochondrial calcium exchange. And so they're constantly looking for what is the actual mechanism by which lithium drives these effects, and then potentially how does the lithium isotope change that mechanistic behavior. So the molecular mechanisms behind this macroscopic effect still need to be determined as an active area of investigation. Another interesting effect is the effect of xenon. So xenon is a noble gas, and it has 7 stable isotopes. Xenon 129 has a nuclear spin of 1/2. 131 has a spin of 3/2, and then the remainder all have a spin of 0. The thing that's interesting about xenon, a noble gas, is that it also acts as an inhalational anesthetic. So if you inhale it in given concentrations, it will cause you to go unconscious, and is a very stable and a reliable anesthetic. It's not used primarily because it's quite expensive. There's an experiment done several years ago now where they were looking at rats being given xenon anesthesia, where that xenon was purified, different isotopes of xenon, and they found that there was a isotope-dependent effect, such that the ones with 32 and 134 is the ones that are longer, the 131 and 129. You need more anesthesia in order to induce that loss of writing reflex, which is a measure of unconsciousness within the animals. So there seems to be an effect of the nuclear spin on the loss of consciousness in these animal models. However, this has not been replicated to date. I don't know of any other study that has tried to look into this, although I do know it is an active area where people are quite interested. So stay tuned, because I know of a few groups that are trying to look into this at the moment. Then the last area is magnesium. So unlike lithium and xenon, which are exogenous isotopes which you add into the body, magnesium is an important cofactor that we have in our body at all time. Okay, and magnesium is involved in a host of dependent processes in our body, metabolism of energy molecules like ATP and GTP. It's involved in physiological signaling, such as the NMDA receptor. It's involved in DNA replication. It's involved in osteoblast proliferation. There's just a lot of different areas where magnesium is involved as a cofactor ion that are responsible for driving a lot of the metabolism within our body. What is interesting about magnesium is that it has 3 stable isotopes, 2 magnesium-24 and magnesium-26, which have a spin of 0, and magnesium-25, which sits in the middle with a spin of 5/2. So this offers us a unique experimental paradigm, because we both have an important biological ion, that we have a heavy version and a light version that have no spin, and a moderate version in weight that does have a nuclear spin. So if we do experiments with all 3 of these, we can rule out any potential mass-dependent effects. Now, there's a history of magnesium-dependent magnetic field effects in the literature, primarily looking at ATP synthesis or ATP metabolism. This is some of the work done by Anatoly Buchachenko, which showed that the ATP synthesis rate of 2 key phosphokinases depends on the presence of it can be dependent upon the concentration of magnesium-25 in solution. And so if you increase the amount of magnesium-25, you can increase the production of ATP from these 2 enzymes. It's very interesting results. However, again, there have been issues with reproducibility as well as with interpretation. So one of the interpretations from this work was that magnesium itself was forming a radical pair, but from standard chemistry, the formation of a magnesium radical is very unlikely, especially in the protein environment. And therefore, magnesium itself forming a radical pair has been all but ruled out. However, the presence of a magnesium spin could still affect another nearby radical being formed in some sort of biochemical process. I will note that this work, the one that is unable to reproduce the Buchachenko work, used a different method of detecting ATP than what Buchachenko did. One was using a radio ligand, one was using a luciferase assay, and therefore, again, I'm urging that when we're doing these types of experiments, we need to make sure that we're comparing apples to apples and not apples to oranges. Okay, so trying to reproduce these effects in the exact same experimental setups, I think, is key and ideal. However, this area, because magnesium is so important in biology, it piqued our interest a few years ago. So with collaborators down in Florida, Rob Smith's group in Florida, and out in Calgary, Christoph Simon's group, we thought it would be interesting to look at specifically the role of magnesium and magnesium isotopes in microtubule polymerization. So I had mentioned before, in neurodegeneration, one of the key proteins that falls apart is microtubules. So microtubules stick together end to end. They're made up of this protein tubulin that stick together end to end and form these nice long strands called protofilaments, which wrap around to form this hollow tube called a microtubule. And these microtubules hold up the processes of the brain. They hold up the dendrites and they hold up the axons. And in neurodegenerative diseases like Parkinson's and Alzheimer's, they fall apart. Now, that process by which tubulin polymerizes into microtubules is dependent on GTP. So it's an analog of ATP. And what happens is that GTP tubulin will stick to the end of a microtubule, causing it to grow. Now, over time, that GTP, in the presence of a magnesium ion, will become hydrolyzed, and a phosphate group will be removed on the end of the GTP, leaving behind GDP. And what that does is it causes tubulin to bend slightly, and it puts a little bit of strain on the microtubule. And so that's what this gray tubulin is here. Now, as long as there is a cap of GTP containing tubulin on the end, the microtubule will stay nice and stable and will not fall apart. However, if all of that GTP tubulin is hydrolyzed, eventually the microtubule will fall apart in a process known as catastrophe. If you then add in GTP tubulin again, the microtubule can be rescued and it can regrow. And this is a process known as dynamic instability. Overall, because this process is highly dependent on magnesium, we thought it would be interesting to change out the isotope of magnesium and see what happened in the absence of presence of a magnetic field. Primarily because it's also been shown that microtubules are dependent on weak magnetic fields, such that if you grow microtubules in a hypomagnetic environment, they will form aberrantly. They will not form properly in a no field environment whatsoever. They need at least the strength of the Earth's magnetic field in order to form in a standard manner. I think I had these slides mixed up, but here is what the microtubules do within a neuron. So here they're forming these bundles in axons and in dendrites. They also form the axonal growth cone and hold up the processes of the axonal growth cone. And so they're very important for overall neural function, as well as synaptic plasticity, the transport of cargo throughout the cell, and therefore it has direct bearing on neuron health and something we can target for a potential treatment in things like neurodegenerative diseases. So what we did is we ordered very, very highly enriched magnesium isotopes. We had natural magnesium, which is primarily magnesium 24, highly enriched magnesium 25, and highly enriched magnesium 26. And then we polymerized tubulins inside a Helmholtz coil to generate a uniform magnetic field. Now, because tubulin is also very sensitive to things like pH, temperature, and so forth, we had to make sure that we were preparing everything the exact same. So my collaborator, Rob Smith in Florida, took painstaking care to make sure all of the buffer solutions were prepared in the exact same manner and had nearly identical isotope concentrations. By isotope concentration, I mean impurity concentrations very close to one another, as well as my master's student at the time, Caleb Siguenza. He made sure that we were placing the wells of the tubulin polymerization far enough away from the magnetic field coils such that there would be no temperature-dependent effects on the system. We then measured the polymerization of tubulin using an optical density-based measure, where we shine light through the system, and on the amount of light that's being scattered, correlating that with the amount of tubulin that's polymerized into microtubules. And so this is what we found. We normalized all of our data to the field off condition for magnesium-26. Magnesium-26 has no nuclear spin, so we surmised that that would be our no effect situation, whereas magnesium-25 does have a nuclear spin, and natural magnesium has 10% abundance of magnesium-25 in it. We then analyzed this, and what we found is that for all conditions, there was no statistical change between any of the conditions, except for the condition where magnesium-25 was present in the presence of a 3 millitesla magnetic field. And we found a gross increase in the amount of overall tubulin polymerized by the end of the experiment. Therefore, we rule out that it's due to some sort of mass effect, because both the heavier and lighter versions of the magnesium did not cause an effect. We determined that it is a magnetic field dependent effect because we only see it when the magnetic field is applied. And we also determined that it is dependent on the isotope. So it is both a combined magnetic field effect and nuclear isotope effect. Since that time, this has been published in Science Advances, but during that review process, what someone had brought up was it's standard when talking about magnetic field effects in biology is that to rule if it is or is not a radical pair mechanism, that we would want to produce a Mary curve, which is a magnetic, the effect of the magnetic field over increasing magnetic fields over a large range. So we can see that trace that Dr. Jones was showing us earlier this morning. At the time, we did not have the money to do this. These isotopes are very, very expensive. They run on average about $2,000 to $4,000 for 50 milligrams. And so these original results, we were only able to do this one experiment. Then I moved away from Florida and moved up to Waterloo, and we are just starting to get our wet lab up and running. And so these are our very preliminary results with an alternate magnetic field. And we see that we're reproducing that effect, at least for magnesium 25. It's a significant effect. We do get a large spread in the natural magnesium, and it's something we're looking into. But again, this is preliminary results on a new setup. So expect more from our group as we move along with this. This seems quite promising. Working with our collaborators at the University of Calgary, we were trying to come up with a mechanism that could describe this. Christoph Simon and Dr. Hadi Zedehegigi are experts in modeling radical pair mechanisms. And so they developed a radical pair dependent model of tubulin polymerization, which they ran both in the presence and absence of different isotopes of magnesium, as well as in the presence and absence of a magnetic field. And they found that our results are consistent with the potential of a radical pair being involved in tubulin polymerization. We do not know yet what that radical pair could be. This is one plausible explanation, but we don't know what the radical pair is. We don't think it's due to magnesium. We don't think it's due to the GTP hydrolysis itself. It may be due to the presence of ambient reactive oxygen species, but this is something we're actively looking at trying to rule out. Okay, so in summary, the radical pair mechanism is a leading candidate to explain weak magnetic fields in biology. This is both from weak magnetic fields as well as nuclear spin effects. It's quite well established for flavin in cryptochrome and for avian magnetoreception, although there is plenty of open questions in that area that people are still investigating. There is some evidence for weak magnetic field effects in things like circadian rhythm via cryptochrome, as well as cell behaviors via cryptochrome, as well as also other cell behaviors based on the presence of reactive oxygen species. Nuclear isotope effects have been observed for things like lithium, xenon, and magnesium, but there are other key isotopes in biology that I think are worth exploring. One is zinc. Zinc also has this same formation of heavy, moderate, and light, where the moderate isotope has a nuclear spin and the other two have zero nuclear spin. Same goes for calcium. So these are other areas that people could be exploring, looking for potential nuclear isotope effects. It seems that the combination of weak fields and nuclear isotopes allows for an added control, at least in certain situations, such as microtubules. But it is something worth looking at, that if we are dealing with systems that are magnetic field dependent, if there can be the introduction of a nuclear isotope in key molecules, it may enhance or dampen the effect, and it would be another way to try and study these mechanisms. From a treatment standpoint, if we were to use this, it might be advantageous in that you could take a supplement of a nuclear isotope that is enriched with nuclear spin and then apply a magnetic field to a certain location to allow for both an enhanced effect as well as a more localized effect. So where do we go from here? I would suggest that more research, obviously trying to tease apart these mechanisms. The more we learn about the actual mechanisms, the more we can start to design treatments and diagnostics based on how they work. Lithium is a great candidate. There definitely seems to be some sort of nuclear spin based effect there, but because we don't know what the actual molecular mechanism is, we definitely need to tease that kind of effect apart. Similarly for microtubules, as well as xenon. These are prime candidates for this nuclear spin effect that I think we need to learn more about. And then again, I'm going to just leave on this note. In this area specifically, there are so many different things that can affect your setup that you're going to want to make sure that you make sure you're accounting for everything. One, we should be having defined protocols whenever we're trying to do experiments, such that they can be shared between labs and reproduce effects or ensure that they are not reproducible based on identical protocols. These protocols would account for things like field artifacts due to stray fields by other equipment around, shielding your setups with things like mu metal to get rid of any sort of stray fields, accounting for temperature variations, changes in your concentrations within biological buffers, accounting for impurities within your systems, and so forth. So throughout, this is my list of references. From the slides, you'll see references throughout. If you're looking for these, I can send them along. And finally, I would like to thank my team, many of whom are here, the two Matthews, Leah and Yashin. Thanks for making your way out here. I encourage you to go see their poster presentations and stay tuned for Leah's talk shortly, as well as our funding agencies and my group of collaborators. Thank you very much.